Dear students, welcome back for a discussion on plant pathology unit, which is a part of paper 1. In previous classes we had discussed about a Tikka disease, which is commonly also known as a leaf spot disease of a groundnut. Today's lecture it is totally dedicated for disease cycle and its control measure with reference to leaf spot disease. So as far as concerned with previous class, I will just make a summary of previous discussion. We have studied about causal organism and related symptoms. As usual, we discussed every time whenever it is a plant pathological aspect, host, how it is get affected by causal organism. But in case of a Tikka disease or leaf spot disease, situation is not like that one, because the causal organisms which are involved here, that is totally different one, which indicates two different phases, early as well as a late phase, and therefore it is more often referred as early leaf spot disease as well as a late leaf spot disease. So the causal organisms which are involved in this process we had discussed comparatively, as well as the symptoms which are produced there, they were also discussed comparatively. In today's lecture we will discuss about how this disease is get completed with reference to the action done by pathogen, or is simply called as a causal organism. Very simple thing is that this causal organism, it harbors itself for several seasons in a soil debris or plant debris. Therefore more often it is said as disease cycle is having a inoculum source which is of a soil borne in nature, or is simply called as a pathogen is a soil borne in nature. Another interesting fact about this particular disease is to be known about up to what extent or how long this disease may be get showing its symptom, or it starts to appear on a crop. And to know about this particular aspect, you have to first of all focus on some basic aspects of this particular pathogen. As I had told you that uh, this particular disease is called as a leaf spot disease. Name itself indicates that it may be either early or it may be late symptoms, and therefore this disease it may starts to show appearance of the symptoms, either it may be in case of a early or late leaf spot situation. Accordingly, the time duration which is required, it may be of a 3 to 5 weeks after showing duration. In fact, one interesting fact you have to focus on that one. Although the telomorphic conditions or telomorphic phases of this particular fungus, they are well known, the ascospores, they are not generally regarded as an important source of a primary inoculum. Basically conidia which are get developed over there in this fungal life cycle, they are either developed from the mycelium which are present in a crop debris in the soil, and following early rains, and after that they may be get deposited on the leaves of young groundnut plant by rain splash, or it may be the wind, and then they initiates the disease cycle. So this is very important aspect, how disease cycle it may be get commenced. Whenever we think about a disease cycle, certain predisposing factors, they are very important. That means environmental factor which controls this disease, we have to consider, we have to take into consideration. The temperature condition which is here required for actual disease commencement or harboring or multiplying this fungus is around 25 to 30 degree Celsius, whereas relative humidity which is required here, it is very high. So then only disease cycle may be get started, or it may be get in favor of that particular pathogen's growth. So the given favorable conditions that may be get more often utilized by that particular fungus, and for that purpose, first of all, lesions they may be get developed on plant, but they are having specific sites where certain symptoms or lesions they appears, and out of that one, the oldest leaves, that is a target site, which are very close or near to the uh, you can say that a host tissue, old very close to the ground, and such area of a leaf that may be get leaf or leaflet that may be get targeted by that pathogen. In short, when we say that the fungus or pathogen is targeting to the host plant, always remember oldest leaves which are very close or near to the soil surface, they are the target of a conidia which lands over there, and of course the landing it is carried out by means of either decimating agent like as a wind, rain splash, or it may be the certain sometimes insects that may be bring these spores or conidia in contact with a leaf surface which is very close to the ground. Okay, and after that what happen, this particular fungal infection it starts over there, or that fungus it initiates its colonization, and later on that may be get spread to the adjoining or nearest plants also, or it may be the nearest leaves also. So it is very very important to remember that the given favorable conditions and which will help to progress this disease particularly from that particular season, or it maybe results into the nearly total defoliation of the plants also, means more often depending upon the intensity or severity of that disease, the leaf which is leaves which are present over there, there is photosynthetic area may be get reduced, and ultimately that defoliation may be takes place, which results into the loss in a crop yield also. Another part which have to remember is that the remnants of this pathogen, they may be as a part of a debris of shed leaves which were infected earlier, and that remains or that harbors for longer time uh, in that particular crop or field. The pathogen that may be survive season to season on volunteer ground, always remember, and infected crop debris also. So another interesting fact which you have to remember is that there is no authentic host species they are known outside the genus Arachis. Okay, means there are different species of Arachis, but genera other than the genus Arachis, this particular pathogen, that is Cercospora Arachidicola, or it may be the Cercospora personatum, that cannot be infect to any other genus. So this is very very important fact we have to remember, or you have to notice this one. So this fact is very very important. Now if you look at another interesting fact is that how this disease maybe get spread at a distant place. That means long distance distribution, is it possible in this particular case or not? Yes, of course it may be possible, and it is by means of a airborne, that's why sometimes it is called as a airborne conidial dissemination process. That means by movement of infected crop debris, or by means of a movement of a infected pods or seeds that are having surface contamination with conidia, or it may be the infected crop debris also. So there are no evidences of either pathogen is being internally seed-borne or not. So such evidences are lacking in the field of this particular area. So if you look for the disease cycle, these were some interesting fact, and if you look at this diagrammatic presentation, you will come to know that how disease cycle may be get takes place. So to know about this entire fact, first of all focus on this diagram, which is related with a first symptom, that is early leaf spot disease of a peanut or groundnut. Here, as far as concerned with a healthy plant, okay, so this healthy plant, as per our previous discussion, it is get infected by means of a inoculum or source, and these sources they are nothing but of course the conidia. So conidia by different ways they come in contact with a leaf surface which is very close to the ground surface. Okay, older leaves which are very close to the ground, they are always get invaded by means of a conidia. Now you know what is causal organism for early leaf spot disease. That is nothing but Cercospora arachidicola. Their conidia, they lands on the surface, and once they lands over there, by means of a natural opening like as a stomata, the infection progresses over there. That means conidia, they germinate, they progress toward the inner tissue that is below to the epidermis, and that spaces which are present over there, then in that space the mycelium, it starts to develop itself or colonization over there. Here you have to focus on this particular structure of mycelial progress. Here there is no as such a hosterial development takes place in case of early leaf spot disease, which is caused by Cercospora arachidicola. And as a result of that, later on this mycelium progresses gradually, and externally symptoms starts to appear, which we had discussed in previous section. Means from the leaf surface, early phases, that's leaf spots or leaf lesions, they starts to appear. Now this is the time where the depending upon the intensity or frequency of such type of colonization or of mycelium or mycelial invasion, the number of leaf spots appears there, or their dimensions or size and number, that may be varied. Of course, this particular situation is under control of predisposing factor what we had discussed, that is temperature as well as the relative humidity. So such infected leaves or leaflets, they may be get fell down and in the form of a debris that may be get accumulated into the soil, and there are two different ways by which this life cycle may be get further continued. Now as it is detached from the plant, the debris which are present over there, they may be get overwintering, or in overwintering stage in a crop residues, and that produces the some special structures which is generally referred as the perithecia. But basically these are nothing but the pseudo perithecal structures which consist of the ascospores present there within a asci. We had discussed about the structure and dimensions of perithecal consisting ascospores as well as within protecting sacs, that is asci. So the ascospores, they are get produced here within the assay, and these are the actual source of a now you can say that a disease causing factor. At the same time, those leaves which are get infected over there, within them they can develop that mycelium that produces some special structures over there, cushion like structures, and these structures they bears or these stalk like structures they bears uh, multicellular, or you can say that is septate spores, and these septate spores are referred as a conidia spores. So or is simply called as a conidia. So these conidia, they may be get detached and become as a inoculum or natural source, and by means of a natural agents like as a rain splashes, or it may be the wind, or it may be by insects, they may be get transmitted from one place to the other, and in this way disease cycle is get completed. So this part what we are discussing is nothing but disease cycle with reference to early leaf spot disease. Similar is the thing about late leaf spot disease. As you know, late leaf spot disease of a groundnut is caused due to the Pioisoariopsis personata, or sometimes it is also called as a Cercosporidium personatum or Cercospora personata. Here basically things are exactly same. That is, there should be a source of a plant, that is host. There should be proper environmental condition, that is predisposing factor like as a room, like as a temperature, and relative humidity should be favorable in that pathogens. So one source is there. There are two sources now. That is, either it may be the conidia or it may be the ascospores. Now how conidia and ascospores are produced? That is a part of a life cycle. So the source of a pathogen or inoculum, it lands on the surface, just we had discussed in case of a early leaf spot through the stomatal opening, that penetrate over here, and here it makes a difference in comparison with a early leaf spot disease. What happens here? In between that host tissue which is present below the epidermis or hypodermal region, you can simply call it as of that leaf. In that space, what happens? Intercellular spaces, they starts to produce the hostoria. All of you know what is the function of hostoria. They are not, branch structures and provides more surface area for absorption of nutrients and food. As a result of it, what happen, this fungus, it occupies the spaces which are intercellular spaces actually, or by proliferating into the mycelium. Mycelium, it proliferates there, it produces hostoria, and in this way the mode of spread or colonization, it is varied in comparison with a late and early leaf spot disease. The rest of the things are same symptoms, they appears externally. Now once intensity is more, or that leaf surface photosynthetic areas get reduced, these leaves are get defoliated, they becomes a part of a debris or crop residues, and in that crop residues the fungus, it further take another in growth depending upon the environmental condition, and accordingly it produces a perithecia which bears the asci, and within that asci ascospores are present. These ascospores liberate out and continues for the further, what is that, host contamination or host invasion. In this way cycle continues with the help of ascospores. Further, in another route, what happens, these infected leaves, this produces the fungal mycelium, and from that fungal mycelia the conidia 4, they are get developing, and multi septate, the conidia, they may be get established there, and these conidia, they are get dispersed by means of different agents like as a rain splashes, or it may be the wind or insect, and they are carried toward the healthy host. Now one point is very clear, this is maybe this is cycle is under control of certain environmental conditions. Now if we are well acquainted about the environmental condition, it is not that much difficult for us to understand what is exactly control strategy there, because any factor which is responsible for a growth of a pathogen, if it is very well known, we can block that factor, that may in nothing but prevention or control of that particular disease. So it is well understood that there are different ways by which we can prevent a disease or control the disease. One of them is a cultural measures for control of a leaf spot disease. Second one, it may be a control by means of a application of certain fungicides. Another method which is more frequently utilized for control of leaf spot disease, there is use or application of cultivars which are highly disease resistant to that particular disease. And another one, but very important aspect, there is nothing but we have to go for biological control measure, and ultimately overall whatever may be measures we know, if they are utilized integratedly, there is nothing but an integrated control of a leaf spot disease. So many strategies are being get utilized for control of Tikka disease, or is simply called as a leaf spot disease of a groundnut crop. So as far as concerned with this particular crop, that is groundnut, certain things are very well for our mind. And first of that one, in what way we are cultivating this particular crop, that itself help us to prevent or control a disease. And out of that first one, there is a you can say that a cultural measures for control of a leaf spot disease. Please keep in your mind, where possible there should be a distinct break in time between successive groundnut crops. As the diseases are largely soil borne, rotation with other crop is obviously very important. So this point is very essential. Plant debris should be removed from the field after harvest. They should be burned in situ. The feed to animal or deep buried. So volunteer groundnut plants and groundnut keepers should be eradicated. This is very important in case of a crop rotation. Depending upon the length of a growing season and cultivars which are to be get grown there, the time of sowing may be adjusted to avoid infection of the crop from outside sources, and to avoid environmental conditions conducive to disease buildup. This is essential. Weeds should be kept under control because their heavy growth may encourage disease development through modification of crop microclimatic conditions. So this microclimatic condition should be get prevented, and as a result of that, what will happen? We will successfully prevent the disease infection. So cultural measures control with fungicides, breeding of resistant cultivars, biological control, and integrated control. This part we are now discussing. Out of that, culture measures, this part is in hand of a farmer or cultivar, because he knows the environmental conditions how suitable are there. Now very important part is about how we can control this disease by using the fungicides. All of you know the fungicidal control of a leaf spot is very effective and economic when they are used by farmers in agriculturally advanced countries where it has been widely adopted. You should know that fungicides are applied by various kinds of tractor propelled machines, or that should be with fixed wing aircraft, helicopters, and more recently through the sprinkler irrigation systems in more developed or advanced countries. The dust formulations that is also get utilized, which consist of various fungicides, means that dust application it is possible by using certain elements, basic elements, fungicidal elements like as a copper, sulfur, or copper plus sulfur. They were more commonly utilized fungicides during 1960s. Apart from that, there are some other commonly used fungicides also. So you have to be keep in your mind the doses of a dusting category of a fungicides, that they are always in higher quantities, and that's higher quantity it leads to the more expenses for the farmer, and now and therefore in case of a developing country it may be very difficult for the farmers also. So this particular method was much more popular in early 90s or 1960s, that time. But later on, the formulations were designed in such a way that spray was also possible, and therefore the spraying fungicides like as a Bordeaux mixture and dithiocarbamate or manab or mancozebs, they were developed and become much more popular. Nowadays, especially during 1970s, what happened? The benomyl or chlorothanoyl and phentine hydroxide, they becomes much more widely utilized. So this is all about different category of a fungicides. Now as we are discussing about this particular leaf spot of a pathogen can be controlled by using some effective methods, what I had just now discussed, that is dust formulations, or it will be the spray fungicides, and how they were get developed or evolved. This particular part we had discussed in more detail. Now nowadays benomyl is very well earlier it was benomyl was very well utilized, but nowadays it is rarely used, because as a leaf spot control chemical alone it is not possible, but along with some other protective fungicides, if it is formulated or if it is used, then it shows some effective uses or effective measures. So this is very interesting fact about a fungicide which is being get utilized by groundnut cultivating farmers. Another part which have to focus here, that is uh, breeding of certain, say that cultivars or varieties. So uh, prior to that, this last point it is very important. Okay, chlorothalonil is nowadays most widely used leaf spot fungicide in United States of America. Let's discuss about a breeding procedure. Okay, so breeding cultivars resistant to the leaf spot, this is one of the essential process, because it is believed that the breeding resistant cultivars is one of the best mean of reducing crop yield losses which takes place due to a fungal pathogen caused diseases. It is one of the important strategy particularly well suited to help small scale farmers of the semi-arid tropic who generally lacks the financial resources and technical expertise which is always required to use chemical control method effectively. So there is also a need of a breeding resistant cultivars or variety in developed countries to reduce farmers' dependence on fungicides and thus bring down the cost of groundnut cultivation. So, to reduce the cost of a groundnut cultivation, the breeding procedure or breeding disease resistant cultivars, that is very important. During 1985 there were no agronomically acceptable groundnut cultivars were found which were having a disease resistant capacity to this Tikka disease or leaf spot disease. But in after that, what happens, screening of groundnut germplasm and their accessions, that was takes place very rapidly, intensively. Research work was carried out in different parts of the world. So what happened, uh, at ECRISAT, okay, uh, you should know the ECRISAT, this is one of the important research center. uh, there were different especially 9 point disease scale uh, procedure was used for screening the germplasm accession and breeding process of groundnut, especially for leaf spot resistance was carried out. So there were various research institutes involved in this entire process of disease control, and through that uh, continuous effort, one variety was get released by University of Florida, United States of America, which is having high yielding groundnut variety and named as Southern Runner EF80202. So this was very effective variety against the leaf spot disease. Again, interesting fact is that yeah, there is a considerable emphasis has been given on screening of wild races, or is simply called as wild species of a arachis or groundnut species for disease resistance of leaf spot. Now this particular data, it provide you the information about how uh, efforts they were made by the ECRISAT. Now I would forgotten to tell you about what is ECRISAT. It is nothing but International Crop Research Institute for Semi-Arid Tropic. So in India, it is at Hyderabad, that is in Andhra Pradesh. It is having the center, that is International Crop Research Institute for Semi-Arid Tropic. And in that particular, like that one, throughout the world there are different centers, and these centers, they are dedicated for various crops which may be get cultivated at arid zone. One of them is a groundnut. By considering various aspects, that how much defoliation maybe takes place under in different species of arachis, that was studied or evaluated. What is the dimension of a lesions, or in what way sporulation maybe takes place. So by considering all these aspects, the disease resistance breeding program was get initiated. So by using this breeding procedure, new varieties are always get utilized over there. Another method of for disease control, that is nothing but a biological control measure, and here the mycoparasites, they are get utilized. Now you should know what is mycoparasite. Mycoparasites, they are nothing but certain fungi which feeds upon another fungi, that is called as a mycoparasite. Means one fungus is an enemy for the another fungus, so that is referred as a mycoparasite. And popular example is nothing but a Dicima pulvinata. So this Dicima pulvinata is also known as Hansfordia pulvinata. So this particular fungus, along with another one that is called as a Verticillium lacanae, this fungus, that is Verticillium lacanae, or nowadays it is referred as the Lacanicillium lacani. So this particular fungus, these two fungi, they are proved as best mycoparasite to control the Cercospora or Phaeoisoreopsis. So if you look at this particular diagram, basically the Verticillium lacani, that is nothing but a entomophagous fungus also. Means here if you look, this is nothing but a nymph body, and on this nymph body cottony structure that is get grown, and it is nothing but the Lacanicillium lacini. That fungus, it grows there, and it invades that insects also. Same fungus culture, it is utilized for control of a leaf spot disease of or Tikka disease also. But if you look for this application of these two fungi, it was observed that they were found much more effective to control this groundnut disease, leaf spot disease, under greenhouse trials or greenhouse studies. However, no serious attempts they were made to use them in a field level. So this is very important. That application should be at a mass level or mass scale level. Otherwise, that entire research may be get failed. Another but last but not least and important one is integrated control of a leaf spot disease. On this you have to focus very carefully, because every effort should be made to utilize all available and compatible disease control measures. Whatever maybe we know, all strategies should be get applied. That is nothing but integrated control of a leaf spot disease. Breeders should always endeavor to combine the leaf spot resistance with the resistance to some other diseases of a groundnut like as a rust. Also, if fungicides are to be get applied, this should be capable to control the leaf spot disease of a groundnut as well as a rust, and possibly applying the fungicide should be combined with some other insecticides which should be considered as insect pest that maybe also further causes certain problem. Means whatever be efforts are possible to control the disease, that all should be utilized together. So that is nothing but a your integrated control of leaf spot disease. So these are the certain references, and as a part of this entire discussion of a plant pathology unit, the first subtopic was the disease known as Tikka disease caused by the Cercospora. So far, what we have discussed here, there is nothing but what is a causal organism, in what way symptoms may be get compared with early and late leaf spot category, and based on this, uh, we had discussed about the disease cycle. Followed by that, we came across with one important aspect, and that is nothing but uh, control measure and preventive measure. As far as concerned with this particular disease, you have to remember some technical things or technical detail, and these are nothing but the comparative symptom formation, and second one, the causal organisms, their differences, morphological differences. So thank you very much for your kind patience.